Welcome to Chapter 3, Ocular Immune Responses, within our series on uveitis and ocular inflammation. Over the next set of slides, we'll build a systematic picture of how the eye handles immune challenges, not as a single organ, but as a collection of distinct immunologic neighborhoods, each with its own rules. We'll move front to back, starting with the conjunctiva and cornea at the ocular surface, then into the anterior chamber, uvea and vitreous, and finally to the retina, retinal pigment epithelium in choroid. Along the way, we'll anchor these concepts to problems you already manage clinically: corneal graft rejection, uveitis, and the newer world of retinal gene therapy. The unifying theme is immune privilege, how the eye protects irreplaceable tissue from the collateral damage of its own defenses. Let's begin with the big picture. Before we get into specifics, here are the 4 ideas that organize everything that follows. First, immune responses in the eye are regional. The same antigen can provoke very different reactions depending on where in the eye it lands, and that regional behavior drives ocular pathology. Second, the eye maintains a specialized internal environment through multiple overlapping immunoregulatory mechanisms, and together these produce what we call ocular immune privilege. Third, one mechanism deserves special attention: anterior chamber-associated immune deviation, or ACAID, which we'll return to repeatedly as the best characterized example of privilege in action. And fourth, this isn't just academic. Understanding ocular immunity is what made modern corneal transplantation so successful and is now shaping retinal gene therapy and cell-based treatments. Keep these 4 threads in mind. Every microenvironment we discuss connects back to them. Let's define regional immunity because it's the conceptual backbone of this chapter. Regional immunity describes how a specific organ or tissue modifies the classic immune response to suit its local needs. Those modifications can occur at any of the 3 phases of an immune response. The afferent phase is antigen capture and transport, getting the antigen to where it can be recognized. The processing phase is antigen presentation and lymphocyte activation, and the effector phase is the actual execution of the response. A tissue can dial any of these up or down. What determines that behavior is the local immunologic microenvironment, the particular mix of cells, barriers, and soluble factors present. The key point for the eye is that it isn't one microenvironment, but several distinct ones packed closely together. That regional diversity is exactly why we study the eye compartment by compartment. Now, the central concept, immune privilege. This term refers to the anatomic, physiological, and immunological adaptations that reduce inflammation and shield vital tissues from immune-mediated injury. The eye isn't alone here. The classically immune-privileged sites include the central nervous system, the eye, the testis and ovary, the pregnant uterus, the hair follicle, and the adrenal cortex. What these share is that inflammation in them carries an unacceptably high cost, so evolution favored restraint. Ocular immune privilege is a defining feature of the eye's internal environment and will come up on nearly every slide from here on. Historically, the concept dates to the 1940s, when investigators noticed something striking. Foreign antigens placed into the anterior chamber frequently failed to trigger the inflammation you'd expect elsewhere. That observation launched the entire field, and it's the seed of ACAID, which we'll develop later. So, how does the eye actually achieve immune privilege? Think of it as 3 layers working together. The first is physical barriers: a partial blood-ocular barrier, a blood-retina barrier, and notably a limited or absent conventional efferent lymphatic drainage, so antigens can't easily traffic out to lymph nodes the usual way. The second layer is an inhibitory microenvironment, built from cell-bound immunoregulatory molecules displayed on ocular cells and soluble immunosuppressive factors dissolved in the ocular fluids. And the third is anterior chamber-associated immune deviation, ACAID, an active immunoregulatory program that suppresses antigen-specific inflammatory responses against antigens encountered inside the eye. Notice these mechanisms are complementary, not redundant. Barriers limit access,
The microenvironment restrains cells that do get in, and ACAID reshapes the systemic response. This framework will recur as we tour each compartment. We start at the ocular surface with the conjunctiva.
We'll begin our regional tour at the very front of the eye with the conjunctiva. As we go through this first section, I want you to hold a contrast in mind. The conjunctiva sits at the opposite end of the immune-privileged spectrum from the internal eye. Where the anterior chamber and retina are immunosuppressive and quiet, the conjunctiva is an active, responsive mucosal surface, well-vascularized, well-drained, and richly populated with immune cells. It behaves much more like the gut or airway liming than like privileged tissue. Understanding why the surface is immunologically active while the interior is privileged sets up the entire logic of ocular immunity. So as we cover conjunctival microenvironments, effector mechanisms, and regulation over the next several slides, keep asking, how does this surface stay vigilant against pathogens while still avoiding destructive inflammation? Let's look at its microenvironment. The conjunctiva functions much like other mucosal immune tissues throughout the body, and its anatomy tells you why. It's highly vascularized, giving immune cells and mediators easy access, and it has effective lymphatic drainage to the preauricular and submandibular lymph nodes, a genuine efferent pathway, in sharp contrast to the lymphatic pore interior of the eye. It's also well stocked with potential antigen-presenting cells. Langerhans cells, other dendritic cells, and macrophages are all present. Put those features together, and you have a tissue built for surveillance. This anatomy allows the conjunctiva to rapidly detect an antigen at the ocular surface, process it, transport it to draining lymph nodes, and mount a response. That's the opposite of immune privilege. This is immune readiness. As we saw in the section intro, this active posture is appropriate for a surface constantly exposed to the outside world. Next, we'll see how it organizes that processing. When the ocular surface is infected or inflamed, the conjunctiva can respond by enlarging its follicles, the follicular reaction you recognize at the slit map. These follicles aren't just swelling. They're organized immune structures containing T lymphocytes, B lymphocytes, and antigen-presenting cells. A useful analogy is the Peyer patches of the intestine. Like those, conjunctival follicles likely serve as sites of localized antigen processing, sampling what's on the surface and initiating responses. Antigens are thought to reach these processing sites by passing through the thin, overlying conjunctival epithelium, which is permeable enough to allow that sampling. This also connects to systemic disease. Conjunctival nodules can appear in conditions like sarcoidosis, reminding you that the conjunctiva participates in body-wide inflammatory processes. So the follicle is the conjunctiva's inductive machinery, the place where surface antigen meets the adaptive immune system. Now, let's turn to how it actually executes a response. Having covered how the conjunctiva senses and processes antigen, let's look at its effector toolkit. The substantia propria is packed with immune effector cells, and mast cells are especially abundant here, a detail worth remembering because it explains the conjunctiva's dramatic role in allergy. On the humoral side, all major antibody classes are represented at the ocular surface. IgA predominates in tears, consistent with the conjunctiva's identity as a mucosal tissue. And those tear antibodies come from two sources: local production within the ocular surface tissues and passive leakage from the circulation. Innate immunity is present too, particularly components of the complement system, which provide rapid, antibody-independent defense. So the conjunctiva pairs a mucosal antibody system with abundant mast cells in complement, a versatile arsenal. That heavy mast cell presence sets up the clinically important response we'll cover on the next slide. Pulling the surface story together, the conjunctiva can support a genuinely broad range of immune responses, both innate and adaptive. On the adaptive side, that includes antibody-mediated immunity and both T and B lymphocyte-mediated responses, the full repertoire you'd expect from a competent mucosal tissue. But one pathway stands out as especially common and clinically important, and it follows directly from all those mast cells we just discussed: IgE-mediated mast cell degranulation. When IgE bound to mast cells encounters its allergen, the cells release histamine and other mediators. producing the itching, redness, and chemosis of allergic eye disease. This single pathway is central to the whole family of allergic conjunctival disorders you'll treat, from seasonal allergic conjunctivitis to vernal and atopic keratoconjunctivitis. So, the conjunctiva's immunological richness is a double-edged sword, excellent defense, but also a frequent site of hypersensitivity. Next, how the conjunctiva regulates and organizes all this activity. To regulate all that activity, the conjunctiva relies on organized lymphoid tissue. Within the substantia propria sits conjunctiva-associated lymphoid tissue, or CALT, the principal site where T lymphocytes are activated within the broader ocular mucosal immune system, abbreviated OMUS. OMUS is a network, not a single structure. It includes the lacrimal gland, the conjunctival epithelium, the corneal epithelium, and the regional draining lymph nodes, all working as an integrated unit to protect the ocular surface. Importantly, CALT and OMUS belong to the body's larger mucosal immune system, which is anatomically and functionally distinct from the mainly blood-borne systemic immune compartment. The mucosal system has its own trafficking rules and its own IgA-dominated character. Both innate and adaptive immunity contribute here to maintaining normal homeostasis at the mucosal surface. So think of CALT as the local command center and OMUS as the regional network it belongs to. Next, we'll see how that network is spatially organized. OMUS has a clean organizational logic, split into two functional zones. There are mucosal inductive sites, where immune responses are initiated, and mucosal effector sites, where the activated cells actually do their work. And these are physically separate, which is the key concept here. The sequence works like this. Antigen-presenting cells at the surface capture and process a pathogen or allergen, then carry it to the local lymph nodes, typically the cervical, preauricular, and submandibular nodes, where they present it to T lymphocytes. Antigen-specific T cells are generated in those nodes. Those primed T cells then travel back to the ocular mucosal effector sites to carry out the response. This induction here, execution there design is a hallmark of mucosal immunity generally, and it explains how the ocular surface can mount specific, targeted responses. It also sets up a fascinating connection between the eye and the nose, which is our next slide. Here's a clinically underappreciated point. Different mucosal tissues vary in their ability to generate responses at distant mucosal sites, and the eye and nose are tightly linked. The anatomic bridge is the nasolacrimal drainage system, which creates a direct connection between OMIS and nasal-associated lymphoid tissue, or NALT. Because of this, the two systems are considered immunologically connected and functionally interdependent. What happens in one influences the other. The evidence is compelling. Intranasal immunization can stimulate IgA production not only in the nasal mucosa, but also in the salivary glands, and, notably, at the ocular surface. In fact, intranasal antigen exposure may produce a stronger tier IgA response than putting the antigen directly on the eye. This integrated ocular nasal pathway isn't just a curiosity. Recognizing it has supported the development of intranasal immunotherapies for inflammatory dry eye disease. That wraps up the conjunctiva. Now we move one structure inward to the cornea. Having covered the conjunctiva, we move to the cornea, and with it a shift back toward immune privilege. This is a crucial section because the cornea is the classic clinical proof that immune privilege matters. It's why corneal transplantation succeeds where other grafts fail. But the cornea also teaches an important nuance. Privilege here isn't uniform across the tissue. As we'll see, the peripheral cornea, near the limbus, behaves quite differently from the avascular central cornea, and that regional split explains a lot of corneopathology. So as we go through corneal microenvironments, the mechanisms that protect it, and the worked example of graft rejection, keep two questions in mind. What makes the central cornea privileged, and what happens when that privilege breaks down? Let's start by contrasting the peripheral and central corneal environments. The single most important idea about the cornea is that its periphery and center are distinctly different immune environments. In the healthy eye, the blood supply is confined largely to the limbus, and the limbus also contains abundant Langerhans cells, antigen-presenting cells poised to respond. Move inward, and the picture changes completely. The paracentral and central cornea are avascular and normally contain few or no antigen-presenting cells. That vascular and cellular gradient is the anatomic basis of central corneal immune privilege. But it's not fixed. Mild trauma, infection, or inflammatory cytokines such as interleukin-1 can recruit antigen-presenting cells into the central cornea, effectively eroding its privilege status. This regional biology has direct clinical correlates. Immune-mediated disorders, like marginal keratitis, characteristically arise near the vascularized limbus, where the immune machinery is concentrated. So remember, privilege is a property of the center, and it depends on keeping that center avascular and cell poor. Next, the specific immune components present. Let's inventory what's actually present in the cornea, and again, the peripheral to central gradient dominates. Plasma-derived proteins, including complement and IgG, are present at moderate levels in the peripheral cornea, supplied from the nearby limbal vessels. Centrally, immunoglobulin concentrations fall off, with only small amounts of IgM detectable. The corneal cells themselves aren't passive. They produce antimicrobial proteins and local immunoregulatory mediators that contribute to defense. Importantly, the cornea likely does not perform substantial local antigen processing, and resident effector cells are normally sparse or absent, consistent with its privileged, quiet baseline. But quiet doesn't mean defenseless. When chemotactic signals are generated, neutrophils, monocytes, and lymphocytes can rapidly migrate through the corneal stroma to reach a site of injury or infection. So the cornea maintains a low-resting immune profile, but retains the ability to recruit cells quickly when threatened. That recruited cellular traffic produces findings we can see clinically, which is our next slide. This slide connects corneal immunology directly to the slit lamp. During intraocular inflammation, recruited immune cells may adhere to the posterior corneal surface, the endothelium. When they accumulate as focal deposits there, they appear clinically as keratic precipitates, the KPs you grade in every uveitis exam. So, a keratic precipitate is, in essence, a visible collection of inflammatory cells settled on the endothelium. There's a second, more specific pattern worth knowing. In endothelial graft rejection, these inflammatory cells can organize into a linear, advancing front known as a cotodaust line. That line marks the leading edge of the immune attack sweeping across the donor endothelium, and recognizing it is essential for diagnosing rejection early. Both findings remind you that the immunology we're discussing isn't abstract. It presents as signs you interpret daily. With the effector picture in place, let's turn to how the cornea protects itself, beginning with its privilege mechanisms.
Now let's formalize how the cornea achieves privilege, tying together the anatomy we've built up. Corneal immune privilege depends on several complementary mechanisms, and normal limbal anatomy and physiology play a central role. Maintaining corneal avascularity, along with the low density of antigen-presenting cells in the paracentral and central cornea, keeps immune activation to a minimum. That scarcity of both APCs and lymphatic channels specifically weakens the afferent arm of the immune response. Remember our three phases, by limiting both antigen capture and antigen transport out to lymph nodes. On top of that, the absence of central postcapillary venules reduces efficient recruitment of circulating effector cells, weakening the effector arm as well. So the cornea restrains multiple phases at once. But privilege is not absolute, and this is the recurring caveat of the whole chapter. Inflammatory cells and soluble mediators can still enter an avascular cornea when the stimulus is strong enough. Next, the molecular side of that protection.
Beyond anatomy, the cornea is bathed in active anti-inflammatory signals, and this molecular layer is where the aqueous humor becomes important. The corneal endothelium is continuously exposed to immunosuppressive factors dissolved in the aqueous, including transforming growth factor beta, alpha melanocyte stimulating hormone, macrophage migration inhibitory factor, and vasoactive intestinal peptide. Together, these soluble mediators tamp down immune activation right at the endothelial surface. The cornea also defends itself with membrane-bound molecules. Corneal epithelial and endothelial cells express Fas ligand, or Fas-L. Fas-L is elegant. It triggers apoptosis in infiltrating neutrophils and lymphocytes that carry the Fas receptor, essentially instructing invading immune cells to self-destruct before they can cause damage. So you have anatomic barriers, soluble suppressive factors, and cell surface killing molecules, all layered together. These defenses suppress both immune initiation and tissue-damaging effector responses. This rich protective system is exactly what makes the cornea the most successfully transplanted tissue in the body. Our next topic.
Here's where all this corneal immunology pays off clinically. Penetrating keratoplasty has a graft success rate exceeding 90%, and remarkably, that's achieved even without systemic immunomodulatory treatment. No oral immunosuppression, unlike essentially every solid organ transplant. Corneal grafts are accepted far more readily than most other transplanted tissues, and the reason is precisely the immune privilege we've been building up. Several protective mechanisms converge to allow this. There are reduced immune responses against the donor antigens in the first place, thanks to the weakened afferent arm. There's active generation of regulatory T cells, which suppress destructive alloimmune activity, and ACAID-like tolerance we'll expand on shortly. And there's apoptosis of inflammatory cells at the graft-host junction, driven in part by the Fas-L mechanism we just covered. So corneal transplantation is the textbook demonstration of immune privilege at work. But privilege can fail, and understanding when it fails is the next slide.
If privilege explains why corneal grafts survive, then rejection is what happens when that privilege is undermined. So let's look at the factors that promote it. The biggest one is corneal vascularization. When new blood vessels grow into the cornea, they're often accompanied by growth of lymphatic vessels and recruitment of dendritic cells, effectively rebuilding both the afferent and effector arms the privileged cornea normally lacks. Inflammation compounds this by increasing stromal expression of human leukocyte antigen molecules, which is normally kept low, making the graft more visible to the immune system. The donor tissue itself can carry donor-derived antigen-presenting cells into the recipient eye, seeding the response. And critically, disruption of ocular immune privilege, including failure to generate ACAID, raises rejection risk. In experimental models, splenectomy is one maneuver that prevents ACAID induction, underscoring how central that pathway is. So rejection is fundamentally the loss of privilege. Next, how the host's systemic state factors in.
Rejection risk isn't determined only at the graft. The recipient's whole body immune state matters too. Changes in the host's systemic immune status can alter graft tolerance, and there are several reported triggers worth knowing. Corneal rejection has been described following systemic immune checkpoint inhibitor therapy, which makes mechanistic sense since those cancer drugs deliberately release the immune brakes that privilege depends on. It's also been reported after SARS-CoV-2 infection and after recent vaccination, presumably through broad immune activation. Now, an important caveat about how to interpret these. These reports are largely isolated observations, case reports in small series, and they do not establish a causal relationship. So counsel patients thoughtfully, but avoid overstating the risk. The practical takeaway is awareness. If a stable graft suddenly rejects, ask what changed in the patient's systemic immune status recently. That completes the cornea. We now cross into the interior of the eye, the anterior chamber, uvea, and vitreous.
We now move from the ocular surface and cornea into the eye's interior, the anterior chamber, the anterior uvea, and the vitreous. This is the heartland of immune privilege, the very region where the concept was born back in the 1940s with those anterior chamber experiments we mentioned early on. Everything gets more restrictive here. The barriers are tighter, resident immune cells are fewer, and the regulatory machinery is more sophisticated, culminating in ACAID, which will finally develop in full in this section. As we go through the anterior chamber's microenvironment, its blood-ocular barrier, its sparse immune cell population, and its unusual antigen clearance routes, Notice how each feature serves one goal, allowing the eye to recognize antigen while preventing inflammation from destroying delicate, non-regenerating structures. Let's start with the anterior chamber's immune microenvironment. Let's characterize the anterior chamber as an immunologic space. It's a fluid-filled compartment where circulating aqueous humor acts as a communication medium, allowing immune cells to interact with the resident cells of the iris, ciliary body, and corneal endothelium. The defining chemical feature of the aqueous is how protein poor it is. It contains roughly 0.1 to 1% of the protein concentration found in serum. That's a strikingly dilute environment, and it limits the inflammatory machinery, like complement, that depends on plasma proteins. But dilute does not mean inert. Despite that low protein content, normal aqueous is biologically busy, carrying immunoregulatory cytokines, neuropeptides, and complement inhibitors. These soluble mediators actively shape immune activity throughout the anterior segment, generally steering it towards suppression. Recall, these are some of the same factors that protect the corneal endothelium. So, the anterior chamber is a low-protein but highly regulated space. Next, the barrier that maintains this composition. What keeps the aqueous so protein-poor and controlled is the blood-ocular barrier. And in the anterior chamber, that barrier is partial and worth understanding in detail. It has two components with different tightness. First, the vessels within the ciliary body are fenestrated, meaning leaky. They allow plasma macromolecules to enter the ciliary interstitium in a size-dependent way, so smaller molecules reach higher concentrations, while larger ones are more restricted. But then comes the real barrier, tight junctions between the pigmented and non-pigmented ciliary epithelial layers. This epithelial barrier is strong, and it prevents most of those interstitial macromolecules from passing through the ciliary body directly into the aqueous. So the ciliary epithelium, not the vessel wall, is the true gatekeeper here. A limited number of plasma-derived macromolecules can bypass this route entirely and diffuse into the anterior chamber across the anterior iris surface instead. This partial selective barrier is exactly what a privileged compartment needs. Now, who lives in this space?
Let's take a census of the anterior uvea's resident immune cells, because the pattern is very telling. Normal anterior uveal tissue contains only small numbers of T lymphocytes and mast cells. Several cell types you'd expect in active immune tissue are generally absent under normal conditions: B lymphocytes, eosinophils, and neutrophils. Consistent with the protein-poor aqueous we discussed, normal aqueous carries only very low concentrations of IgG and complement components. But there's an important exception to this quiet picture. The iris and ciliary body actually contains substantial numbers of macrophages and dendritic cells. These may function as antigen-presenting or effector cells if needed, though, as with the cornea, significant local antigen processing is thought unlikely. So the resting anterior uvea is deliberately understaffed with inflammatory effectors, yet equipped with sentinel APCs. This raises an obvious question. Without lymphatics, how does the eye clear antigen and talk to the immune system? That's exactly the next slide.
Here's an elegant solution to a real problem. The inner eye lacks clearly defined lymphatic channels, so how does it get rid of antigen and communicate with the immune system? For soluble material, clearance happens mainly through the aqueous outflow pathways, the same trabecular route that drains the eye's fluid. Particulate material has two additional routes: endocytosis by the trabecular meshwork endothelial cells and phagocytosis by macrophages. Now here's the immunologically crucial part. Soluble antigens may remain intact, enter the venous circulation through the outflow pathway, and travel to the spleen. That splenic route is central to ACAID, as we'll see shortly. The takeaway is that even without conventional lymphatics, anterior chamber antigens communicate efficiently with the systemic immune system. The eye simply substitutes a blood-borne, spleen-directed pathway for the usual lymphatic one.
And routing antigen to the spleen rather than to lymph nodes is precisely what produces the deviant, privileged response. Now let's shift posteriorly within this section to the vitreous.
Turning to the vitreous, we enter territory that's less studied than the anterior chamber, but increasingly recognized as biologically active and complex, not the inert gel it was once assumed to be. Proteomic studies reveal that the vitreous contains a wide range of proteins derived from both intraocular and extraocular sources, indicating real immunologic participation. A key contributor is the retina, which is an important source of the inflammatory mediators found within the vitreous, so the vitreous reflects the immune state of the tissue behind it. The gel also has an interesting physical property. It can electrostatically bind charged proteins. That binding capacity lets the vitreous function in two ways, as an antigen reservoir, holding antigen in place, and as a surface for leukocyte adhesion, providing a scaffold where immune cells can gather. So the vitreous is both a chemical archive and a physical platform for immune activity. Its autoimmune potential and resident cells come next. Building on the vitreous as a biologically active tissue, consider its autoimmune potential and its resident immune cells. The vitreous contains type 2 collagen, and that matters because type 2 collagen is an antigen implicated in rheumatoid arthritis. So the vitreous carries a potential autoantigen that could participate in autoimmune processes. The key resident cells are hyalucytes, which are modified resident macrophages living within the vitreous. Hyalucytes do real immunologic work. They contribute to vitreous immune regulation and frequently serve as antigen-presenting cells, much like the sentinel APCs we've met in other compartments. They also respond to cytokine signals and can participate in the pathogenesis of inflammatory and proliferative disorders. The clearest clinical example is proliferative vitreoretinopathy, where hyalucyte activity contributes to the membranes and traction that complicate retinal detachment surgery. So the vitreous has both the antigens and the cellular machinery to drive disease. Now let's step back and ask why intraocular immune regulation has to be so tight. Before we detail ACAID, it's worth pausing on why the eye invests so heavily in immune regulation. The stakes explain the whole design. The core reason is fragility. Even modest inflammation can permanently damage the eye's delicate structures and cause severe, irreversible vision loss. Unlike skin or gut, these tissues don't regenerate, so the eye can't afford the collateral damage that a vigorous immune response causes elsewhere. That's why the anterior chamber, anterior uvea, and vitreous all rely on multiple overlapping regulatory mechanisms that deliberately limit the intensity of any immune response. And this privilege is remarkably broad. Ocular immune privilege has been demonstrated across many different antigen types, including transplant antigens, tumor antigens, haptens and soluble proteins, autoantigens, and bacterial and viral antigens. That breadth tells you privilege is a general strategy, not a response to one particular threat. With that motivation established, we can now examine the best characterized regulatory mechanism of all, ACAID. Here is the mechanism this whole chapter has been building toward: anterior chamber associated immune deviation, ACAID, the best characterized mechanism of ocular immune privilege. The defining observation is that the same antigen produces different responses depending on where you introduce it, which is exactly the regional immunity idea from slide 3, made concrete. Introduce an antigen subcutaneously, and you typically get a strong delayed hypersensitivity response, the classic inflammatory cell-mediated reaction. Introduce that identical antigen into the anterior chamber, and you get something deviant, a strong antibody response, but little or no delayed hypersensitivity. The eye has redirected the response away from the inflammatory tissue-damaging pathway. This depends on that spleen-directed antigen route we discussed a few slides ago. And ACAID is powerful enough that it can even suppress a delayed hypersensitivity response that's already been established elsewhere. So ACAID doesn't ignore antigen, it recognizes it and reshapes the response.
Let's look at precisely which responses it suppresses.
Let's be precise about what ACAID actually does and doesn't do, because the distinction matters clinically. The essential concept is that ACAID weakens the effector phase of immunity rather than eliminating antigen recognition. The eye still sees the antigen, it just restrains the damaging response. The workhorses here are regulatory T cells, which suppress the major cell-mediated pathways. Specifically, they dampen delayed hypersensitivity and cytotoxic T lymphocyte activity, the two arms most capable of destroying ocular tissue. In terms of the helper T cell framework, ACAID inhibits Th1-mediated immunity, the classic driver of delayed hypersensitivity, and it also reduces inflammatory disease driven by Th2 responses, so its suppressive reach extends across both major helper subsets. This is why ACAID is such an effective guardian of privileged tissue. It targets exactly the effector mechanisms that cause collateral damage, while leaving recognition intact. But antibody responses aren't simply switched off. They're modified in a clever way, which is our next slide. So what happens to the antibody response under ACAID? It isn't eliminated, antibody production remains intact, but it's qualitatively redirected toward non-complement fixing antibodies. That's a subtle but important shift. By favoring antibodies that don't activate complement, the eye reduces complement-driven inflammation and lowers the risk of antibody-mediated damage to its own tissues. You still get humoral immunity and its protective functions, but stripped of one of its most destructive effector arms, a recurring theme of protecting the tissue while preserving recognition. One final and essential caveat, consistent with everything we've said about privilege in this chapter, ACAID is not absolute. A sufficiently strong immune stimulus can overwhelm the eye's regulatory environment and break through, which is part of why severe or aggressive intraocular infections and inflammation can still devastate the eye despite all these safeguards. That completes our tour of the anterior chamber, uvea, and vitreous. We now move to the final region, the posterior segment.
Our final section takes us to the back of the eye, the retina, the retinal pigment epithelium, the choriocapillaris, and the choroid. In many ways, this is the frontier of ocular immunology. These environments are less fully defined than the anterior segment, and active research is still mapping them. But the stakes here are the highest, because the neural retina is non-regenerating tissue, where inflammation means permanent vision loss. As we go through the posterior segment's barriers, its surprisingly rich population of antigen-presenting cells, and its own ACAID-like regulatory mechanisms, you'll see the same principles from earlier sections reappear: barriers, sparse effectors, resident sentinel cells, insoluble suppression, adapted to retinal biology. And this section pays off in one of the most exciting clinical arenas in ophthalmology, retinal gene therapy, which we'll use as our closing example. Let's begin with the posterior segment's microenvironment and its barriers.
Let's map the posterior segment's barriers, which follow the same logic we saw anteriorly, but with retinal specifics. First, a candid acknowledgement. The immune environments of the retina, RPE, choriocapillaris, and choroid remain less fully defined than those of the anterior segment. The blood-retina barrier has two parts. The inner blood-retina barrier is created by tight junctions between the retinal vascular endothelial cells. The outer blood-retina barrier is formed by tight junctions between RPE cells, which separate the neural retina from the choroid. So the RPE is both a barrier and, as we'll see, an immunoregulatory hub. Beyond that barrier, the choriocapillaris behaves very differently. Its vessels are highly permeable, allowing many plasma macromolecules to leak into the surrounding choroidal extravascular space. And as elsewhere in the eye, well-developed lymphatic channels are absent from both the retina and choroid. So we have a tightly sealed retina sitting next to a leaky, protein-rich choroid. Next, the cells that watch over this space. Despite that limited lymphatic drainage, the posterior segment is far from immunologically empty. The retina and choroid contain numerous cells capable of antigen presentation. The key players in the retina are the microglia. Retinal microglia are bone marrow-derived cells related to dendritic cells, and they're distributed through the inner retina, from the ganglion cell layer down to the outer plexiform layer, where they act as resident sentinels. In the resting state, they're quiet and ramified, but in response to injury or inflammation, microglia can change shape, become activated, and migrate to the side of trouble, a dynamic behavior you'll hear invoked in many retinal inflammatory and degenerative diseases. The choroid and choriocapillaris, for their part, are rich in macrophages and dendritic cells, consistent with that leaky, plasma-exposed environment we just described. So the posterior segment pairs a sealed barrier with a well-staffed surveillance network. Let's look more closely at the other immune cells within the choroid. Let's complete the choroidal census. The normal choroid contains a moderate number of mast cells, and they cluster particularly around the arterioles, a location that becomes important on the next slide. Lymphocytes are present only in small numbers, while eosinophils and neutrophils are typically absent under normal conditions, the same resting quiet we've seen in privileged compartments throughout the eye. But that baseline changes dramatically with disease. During pathologic or experimental inflammation, the choroid, choriocapillaris, and even the retina can be infiltrated by a full inflammatory cast: T lymphocytes, B lymphocytes, macrophages, and neutrophils. This capacity to recruit heavily is why the choroid can act as a reservoir for immunologically active cells, and clinically it can even develop lymphoid hyperplasia. So the choroid is quiet at rest, but has enormous inflammatory potential in reserve. That perivascular mast cell location I mentioned turns out to play a role in how inflammation spreads, which we'll cover next along with cytokine regulation. Now, let's connect those perivascular mast cells to the regulatory chemistry of the posterior segment. Because choroidal mast cells set around the arterioles, they're well positioned to help facilitate the spread of activated immune cells into other ocular tissues. Their mediators can increase vascular permeability and open the door for cellular traffic. Balancing that pro-inflammatory potential is a suppressive network. The RPE, along with several retinal and choroidal cell types, including pericytes, can produce immunomodulatory cytokines. A prime example is transforming growth factor beta 2, closely related to the TGF-beta we saw suppressing inflammation in the anterior chamber, showing how the eye reuses the same regulatory tools front and back. These locally produced mediators can alter both the intensity and the character of any subsequent immune response, tuning it up or down. So the posterior segment holds pro-inflammatory triggers and suppressive brakes in careful balance. Next, we formalize the posterior segment's own version of immune privilege. Let's pull the posterior segment together as a regulated, privileged system, and note that privilege isn't confined to the anterior chamber. Antigen placed in the subretinal space can trigger an immune-privileged response that looks analogous to ACAID, giving the back of the eye its own deviation mechanism. The iris, ciliary body, and RPE all help maintain ocular immune homeostasis, using both soluble mediators and cell surface regulatory molecules. The RPE is especially versatile. Its soluble factors can suppress activation of antigen-presenting cells, reduce activation of effector T lymphocytes, promote regulatory T cell activity, and stimulate myeloid-derived suppressor cells. 4 distinct breaks from a single tissue. These mechanisms aren't just theoretical. They may critically influence the success and safety of retinal transplantation, stem cell-based treatments, and retinal gene therapy. That said, be honest about the limits of current knowledge. The specific roles of the choroid and choriocapillaris in the afferent and effector phases remain incompletely understood. That therapeutic relevance leads directly to our closing clinical example: retinal gene therapy. We'll close the chapter with retinal gene therapy, the clinical arena where posterior segment immune privilege becomes practically decisive. The basic concept is straightforward. Retinal gene therapy introduces a functional replacement gene into the neural retinal cells, or the RPE, correcting an inherited deficiency at its source. Delivery commonly uses a genetically modified, replication-deficient viral vector, a virus stripped of its ability to replicate but retained as a vehicle to carry the therapeutic gene into the target cells. Once inside, the cells transduced by the vector begin expressing the transferred gene, ideally restoring the missing protein, and with it some visual function. Here's where immunology becomes central. In most other tissues, the immune system recognizes and eliminates the viral vector, and that immune elimination can reduce or even terminate expression of the introduced gene. The privileged eye offers a more hospitable environment for durable expression, which is exactly why the eye became the pioneering organ for gene therapy. Let's look at the first approved product.
The landmark example is Voratine Nepervovax RYZL. In 2017, the FDA approved it as the first gene replacement treatment for a retinal disease, a genuine milestone not just for ophthalmology, but for gene therapy as a whole. It's indicated for retinal dystrophy caused by biallelic RPE65 mutations, which includes certain forms of Leber congenital amaurosis, patients who carry 2 defective copies of the RPE65 gene and suffer severe early vision loss. Mechanistically, it uses an adeno-associated viral vector, an AAV, to deliver a functional copy of RPE65 into the retinal pigment epithelium, where that gene is normally expressed and needed for the visual cycle. And crucially, given everything we've discussed about targeting the privileged posterior segment, it's administered by subretinal injection, placing the vector directly into that immune-privileged subretinal space to favor durable expression and limit immune elimination. This proof of concept opened the door to a whole pipeline of retinal gene therapies, which we'll survey next.
Voratine was the first, but it's really the opening chapter of a much larger story. AAV-based treatments, building on that same adeno-associated viral vector platform, delivered to the privileged retina, are now being investigated for a range of inherited retinal disorders. The current targets include achromatopsia, choroideremia, Leber hereditary optic neuropathy, X-linked retinoschisis, and X-linked retinitis pigmentosa. Notice the breadth here. These span different genes, different affected cell types, and different inheritance patterns, from cone dysfunction to optic neuropathy to progressive rod cone degeneration. What they share is a monogenic basis that makes gene replacement conceptually feasible and a location within the immune-privileged eye that makes durable transgene expression more achievable than it would be almost anywhere else in the body. So the field is expanding rapidly on the foundation Voratine established. But privilege doesn't mean the immune system is absent, and understanding the immune response to these vectors is essential, which brings us to our final content slide. Let's end on an essential reality check that ties the whole chapter together. Ocular immune privilege does not eliminate the immunogenicity of viral vectors. Privilege reduces and reshapes immune responses, as we've stressed repeatedly, but it doesn't abolish them, and the route of delivery matters a great deal. In animal models, intravitreal delivery has produced a systemic antibody response, whereas subretinal delivery has generally produced less humoral immune activation. That difference is a direct, practical application of the compartmental immunology we've studied, and it's a major reason Voratine is given subretinally rather than into the vitreous. In real patients, intraocular inflammation still occurred in roughly 5% of those treated with Voratine Nepervovax RYZL, uncommon but not negligible. To reduce that risk, systemic corticosteroids are started 3 days before treatment, preempting the immune response to the vector. So even in the most privileged tissue, we actively manage immunity. Let's wrap up. That brings us to the end of Chapter 3 on ocular immune responses. Let's recap the arc we traveled. We began with the core concepts of regional immunity and immune privilege, then toured the eye front to back, the immunologically active conjunctiva, the regionally privileged cornea, the tightly regulated anterior chamber, uvea, and vitreous, and finally the emerging immunology of the posterior segment. Throughout, one theme recurred. The eye recognizes antigen, but restrains the damaging effector response, using barriers, sparse effector cells, soluble suppressive factors, and active deviation mechanisms like ACAID to protect irreplaceable tissue. We saw these principles pay off clinically in the success of corneal transplantation and in the pioneering field of retinal gene therapy. Carry forward this central lesson. Understanding where and how the eye modulates immunity is the key to both diagnosing ocular inflammation and designing the next generation of ocular therapies. Thank you.